Yeah, so this is a phase 2 trial in triple-negative metastatic breast cancer. Imprime PGG is a beta glucan agonist, which is an innate activator of the immune system. And in this study, it was used in combination with a common PD-1 drug, pembrolizumab. And the concept here is that in triple-negative breast cancer, single-agent PD-1 therapy has shown very little activity in chemo-refractory metastatic disease with single-digit response rates. And so in this trial, we are using an innate activator of the immune system to activate the myeloid compartment in the tumor microenvironment, as well as the T-cell activation with pembrolizumab to synergize and hopefully produce a more robust immune response. So this is a phase 2 trial. Previously, Imprime PGG had been looked at in a series of phase 1 trials, and a dose and schedule was determined, a weekly schedule. And so we combined that weekly schedule of Imprime PGG with a standard dose of pembrolizumab, and we treated 44 patients with triple-negative chemo-refractory breast cancer. So interestingly, obviously it's a non-comparative trial, but based on significant historical control, we saw improvement on a number of efficacy parameters. The response rate was 16%, which is larger than the historic response rate of around 5% in this population with single-agent PD-1. But even more exciting, the disease control rate beyond 6 months in this heavily pretreated group of patients was 25%. And the median survival was 16.4%. Again, much better than historical single-agent activity. Well, in addition to the overall population of patients that were triple-negative, there was a subgroup of patients that was particularly interesting that were not de novo triple-negative positive, but had previously been hormone receptor positive and had been subjected to a number of endocrine hormonal therapies, and then became sort of triple negative secondarily. This is an interesting subgroup that's not often part of triple negative studies. But in that subgroup, again, only 12 of the 44 patients, but half of them had major responses, and half of these were durable beyond 6 months. So that's a subgroup we really want to look at going forward. But based on these data, both from the overall triple-negative population as well as this secondary triple-negative population, we certainly want to expand this data to larger cohorts of patients. And interestingly, there was translational science, which was also very revealing. Out of the 44 patients, we had blood before and on treatment in these patients in 43 of the 44. And in all those patients, there was evidence when the myeloid compartment or the T-cell compartment in the peripheral blood was activated, that led to the more higher levels of response and efficacy. And then we also had some paired tumor tissue on 4 patients. And in all 4 patients, there was repolarization of the myeloid component, which is what you would hope for with the Imprime PGG component. And then there was also an activation of the CD8 T cells, which you'd expect from the pembrolizumab. So there was proof of concept, not only that there was some preliminary activity here that looked very interesting, but translationally, both in blood and tumor tissue, we saw evidence of mechanism of action of both the innate immune system and the adaptive immune system, which is very encouraging. Well, so the next phase is really, there's nothing, I mean, triple negative is a good disease because single agent PD-1 has a relatively low activity rate, but there's nothing specific about these two immune mechanisms that would be confined to triple negative breast cancer. So obviously we've also looked at this combination in melanoma, a very active immune disease with PD-1 therapy, and we're hoping to explore that further. So we want to look at both immune-sensitive diseases with PD-1 that we might make better with this drug, and then diseases where relatively cold, so to speak, in terms of low responses to T-cell antibodies, and see if we can make cold tumors warm or hot, like we hope we did in this situation.